Hello, I am Jocelyn Auger and I started my British Heart Foundation sponsored Daphne Jackson Fellowship in January this year after a 14-year career break. I have the huge privilege of working at the medical school at Birmingham University. In this video, I'd like to share my research with you. Here is the title of my fellowship research project, which I'd be pleased to chat to you about any time. Essentially, I'm developing a model of a blood vessel to which I can apply some fancy new microscopy techniques. Today, I want to share the first steps I've taken in learning how to persuade endothelial cells to sit nicely in my model. Endothelial cells are the cells that line every blood vessel. They provide a barrier between the flowing blood and the external tissues. In a healthy blood vessel, the blood flows smoothly through the vessel, and there is little interaction of the clot-forming blood cells with the endothelial cells. However, if a vessel is damaged, for example as a result of injury, the endothelial layer is broken and other blood cells called platelets are exposed to the matrix underneath. This activates the platelets to stick together with other cells to form a clot to stop blood loss. This maintenance of blood flow is known as hemostasis and is a good thing. However, a clot can also form inappropriately in a blood vessel in a process called thrombosis, which is a bad thing. This can happen as a result of atherosclerosis, where fatty plaques build up underneath the endothelial cell layer. If the plaque ruptures, the platelets are exposed to the proteins that promote clot formation, leading to a clot forming within a vessel. If this becomes big enough to block the vessel, it can lead to injury or death of the surrounding tissue, which is what happens in a heart attack or stroke. Thrombosis can be modelled in vitro with a flow chamber, where a pump is used to pull blood through a chamber containing a glass cover slip. Work by myself and others has demonstrated thrombus formation when blood is poured over a cover slip coated with substances such as collagen, which is found in the matrix underneath the endothelial cell layer. We can see this under a microscope when we stain the platelets with a dye, as shown here. I now want to incorporate endothelial cells into this model to build on the physiological relevance. This work has been pioneered at the Platelet Research Group in Maastricht, so I took a trip there to learn some of their tricks. I have since developed a novel method of applying endothelial cells to a glass cover slip by pipetting a bubble of cells and allowing surface tension to hold the cells in place. It took me a while to get this right, and I produced some very unhappy endothelial cells along the way, but I'm making progress, and here are some of the pretty images I've created. Here we can see the characteristic cobblestone morphology of endothelial cells, with blue DAPI staining of their nuclei and magenta VE cadherin at cell junctions. This result was something to celebrate. The cover slip can also be slotted into a flow chamber and blood pumped through. And here I show, on the left-hand side, that when blood is flowed over collagen, these green stained platelets formed nice thrombi. Whereas on the right-hand side, in the presence of endothelial cells, which are stained magenta and blue, very few platelets are able to bind. Another result to celebrate. Going ahead, I'm going to use more advanced microscopy techniques to look at the connections between platelets and endothelial cells in more detail. Then I will model some cardiovascular diseases and characterise some antithrombotic drugs. Thank you for listening, and thanks to my Daphne Jackson advisor Sally Bolton, my supervisors Steve Thomas and Natalie Poulter, and to the Daphne Jackson Trust and the British Heart Foundation for facilitating my return to research.